Okay, we're back in business. Let's move on to our second speaker. Speaker before the break, but of course you're free to run out and get coffee anyway.
Don't tell them that.
But not during the talk. So our next speaker is David Kaplan from Tufts University. David's the Stern Family Professor of Engineering and the chair of the Department of Biomedical Engineering at Tufts University, which is a component of the Tufts University School of Engineering. And David is going to keep on the convergence theme and tell us about tissue engineering impacts on cancer studies. David.
Thanks, Phil. Good morning, everyone. Great to be here, as always, except for getting downtown with traffic, but we'll get past that. And thanks, Charlotte, for kicking off everything today, and thanks for the invite to talk to you and tell you a little bit about what we're doing. So my goal is very similar to the theme that Charlotte conveyed, and that is I really want to focus more on the tools, not so much a specific disease phenotype or genotype, although I'll show you a little bit about where we're going with our models. But really my message is pretty simple, and that is with the field and the rapidly evolving field of tissue engineering, I think there are tremendous new opportunities to exploit this technology to really make some progress in understanding and treating cancers as we look ahead. So really I'm just going to give you maybe a little too much philosophy to start off. We think about this, and then I'm just going to quickly go through three of our many tissues we work on, tell you what the tissue is, how we make it a little bit, and then one slide at the end of each tissue about what we're doing on the cancer side, just so you see where we're going. The best outcome today would be someone who wants to come to our lab after I talk and learn what we make and take it back to your lab to use for your disease models. So that would be fantastic, and that's something we do all the time. So please don't hesitate if there's something we're making and trying to understand that can be a benefit to your own research. All right, so this is the big picture philosophy. I think most of you know why 3D tissues are important by now, and I don't want to really have to restate all that. I'll just say that we think about our in vitro systems very, very carefully, as you'll see, and we care a lot about what cells, what combinations of cells. We care about the matrices. You heard a little bit about that. I'll try and take that even to a more detailed level, and we care a lot about the environment. And the point of tissue engineering is really building a system and have all those components talk to each other to then get something that is truly relevant to the human response. Because if it's not, I don't see the value for the kinds of goals this symposium is about. And so we use our systems not just for disease, which is where I'll focus today, but to look at drug effects, look at toxicology, issues of nutrition, and many other things, as I'll show you briefly. So the real value here is getting away from 2D cell cultures as your only metric and only using animal studies on the other side as your only metric and trying to marry the best of all worlds. And so that's why I think 3D tissue culture based on human cells offers something quite unique. And you can read this, I don't want to really repeat it, but if everyone talks about personalized medicine today. Well, personalized medicine can even come in the form of tissue screening tools now. That's the point here, right? You can build these tissues from each one of your own cells through the techniques many of you know already. And as long as you do this properly, you have a system that can be very valuable for many things. And so we care about all this here. I can't cover all this today, but we have systems that we vascularize. We have systems that we innervate. I won't deal with those details, but the point is we're starting to get very, very complex and sophisticated about what we can do with the cells to give us readouts that are relevant. So for our lab, all I care about are these red highlights at the bottom. If it's not human physiological relevant, I don't want to deal with it, right? Number two, I care a lot about not just acute, but also chronic effects in our tissues, and that's really important to me and something that's appreciated, I think, in the field, so I'll harp on that too many times. And as always, feel free to disagree with me. And then the last bit is imaging interfaces, and Charlotte touched on that with the work with Irene, and I'll touch on that just at the end because of time on what we're doing there. Because if you're going to long-term 3D human cultures for the readouts we want for both acute and chronic effects, you can't have tissues that you have to take down, destroy, do immunohisto and all the other things. You need something online that you can image in real time and determine what's happening metabolically, toxicologically, structurally, and so on. So that's where the work with Irene comes in. All right, that's philosophy. The other philosophy is here's the field today, and I do this sort of as developmental biology. So on the left is where the field started 15, 20 years ago with some really nice work that simply said, I should point, I guess, you can take some cells, you can pick your cell type, it didn't matter at that point, take some cells, find some spaghetti with high surface area, which is a biomaterial in fancy terms, put the cells on the spaghetti and have them attached, grow them enough, and then reimplant them in the human body and you have a cure, right? And of course that wouldn't work well. In fact, the only thing that evolved out of the 20 years ago work was all the work on tissue engineered human skin, and that's a commercially viable technology. But if you look at that technology, and organogenesis is one of many fields that sells that to help a lot of patients, it's a very, very simple system. It's not complex by any means, but very successful. But there is no other example of a successful tissue engineered product today. And so what did that mean? That meant the field took a step back and said, what are we doing wrong and what do we need to do better? And over the last 15 years, as you all know, the developments and insights into stem cell biology, sophistication in biomaterials, so not just spaghetti anymore, now we can sculpt and control all the features you need for a biomaterial to make the niche what you want for those cells in that tissue. And we can get really fancy with bioreactors to create the environment around those cells. And now we can do things that 15, 20 years ago you only dreamed about. And that's why I think we're at this sort of revolution now on the right, because we have the tools. Whether we talk about them in the context of disease today, you can use those same tools applied to many other areas of interest in both biology and engineering and so on. And again, anytime happy to talk about these other areas because we do have some ongoing work in some of these as well. So for today, really we're just after how can we propagate those tools towards the study of cancer, and I'll focus only on that with the three systems. Again, the underlying theme here, though, is the right cells, the right matrices, and the right environment. It sounds easy. It's not, as you just heard. You know, you start with Matrigel, and then you go running away from it because it's so complex and undefined. And then you have to rebuild from first principles what you want. And sometimes you guess right, and sometimes you don't. And then ultimately for us, what we're after for our disease models is creating the niche to give you the relevant human responses. We started out with cells thinking we'd be simplistic with one cell type, and it turns out that it's never a good answer. You always want multiple cell types because cell-cell signaling is so important, as many of you know. And then again, to continue to beat on the time issue, we care about chronic more than just acute effects. So two overview slides I like to use a lot when I give these kinds of talks. One is if you look at the field of tissue engineering, and this applies in many areas, you can go from the left to the right at the top in sort of pictorial fashion. You all know how to do 2D cell culture or probably have been exposed to it, and that's sort of the tried and true approach to start. And as you work from the left to the right, you're just adding complexity, right? So you're getting more complex systems to the right. You're dealing with longer ability to cultivate your tissues over time. You're adding physiological complexity, and I would argue you add relevance. Okay, so that's great. Why doesn't everybody do what's on the right-hand side? Because it's very costly, it's very time-consuming, you're prone to contamination, and the students have to graduate somewhere between 3 and 5 years. So what we all do is start at the left and screen, right, and work our way to the right, just like in drug discovery, and there's nothing wrong with that. My message today, though, is don't stop until you get to the right. Okay, because if you stop before you get to the right, you'll get some useful data, but you're not going to get all the kinds of data you can potentially get out of your systems. So our mantra in our group is get to the right, okay? And we don't always get there right away, but we usually do. And a good example is we've published, and again, this is with Irene, on systems that we've grown for 6 months in continuous culture in the laboratory. And if you think about most diseases, most drug effects, most nutritional effects, it's not a short-term effect, right? You're after really slow developing effects. So you need these systems to run for long periods of time. And I know it's a pain, but it's worth thinking about. So I put this into biological terms since I figured most of you are biologists, right? So I used a growth curve. This is more from prokaryotic literature, and you'll probably be familiar with this, right? So A is your bacteria starting to grow, and just think of this as your human cells in your tissues, right? B is your logarithmic growth. The cells are starting to get comfortable and doing what you want them to do in whatever system you set up. C is what we call steady state, right? That's where you want to get your readouts. That's where your tissue is stable, your disease model is stable, and you can start to generate data that may be more relevant to the human condition. And then if you wait too long, D is where things start going south, right? So the cells are starting to die, overgrow, whatever it is, and now the readouts are less relevant. So my last point of emphasis on the chronic versus acute is almost every system people use today is on the left-hand side there, whether it's 2D cell culture, transwells, microfluidics, tissue on a chip. They're all wonderful systems, but they end somewhere around 2 to 3, if you have a really good system, maybe 4 weeks. Great, but just keep in mind you're limiting what you can then use those systems for because they're not steady past a few weeks. And again, if we're looking for long-term effects, we need to get to perfused cultures, and we can go out for months and months at a time. So it's good to keep all that in mind if you're really interested in the kinds of things both this symposium is about, as well as what I mentioned before, nutritional issues, drug effects, and for us also just tissue regeneration. Can we damage our tissues, watch the repair, and learn how to improve those repair modes? Even all in vitro, we learn a lot by doing that. All right, so finally we're done with my philosophy. Let's go on. First tissue will be really quick. I just want to talk about fat. And this will be in the context of what Charlotte already spoke about in terms of breast cancer. But first, the system, because as I said, I want to tell you about the tools, not so much the disease. So we spend a lot of time studying three-dimensional fat tissues. This is led by Roz Abbott as a postdoc in the group. And the simple way to describe it is we get lipoaspirate samples from patients. And then we used to go through the process of isolating single cell types from that lipoaspirate through all the FACS sorting, all the mess you go through, seeding our tissues, generating these tissues. And they look good, but they weren't great. And then we took a step back and just took the whole mess that we get from the patient and we seed that into our biomaterial scaffolds and grow our tissues. And now the results are outstanding. So it's a lesson in how important cell complexity is in your systems. And once you seed and understand what grows well, you can work backwards and figure out what cells like the niche and how to then exploit those kinds of insights. So one is the cell sourcing is important. In the middle is the scaffolding, the biomaterials, the matrix that's going to create the artificial environment in the laboratory, the niche, if you want to call it that. So it doesn't matter so much what we use here. What matters is all the designing that goes into choosing the best system for that given tissue. And it'll be different for every kind of tissue. It's even different if we're growing white adipose tissue as opposed to metabolically active brown adipose tissue. So it's not a one-stop shop here. So I gave you a list of things we sort of have to go through for every new tissue in the lab, and we've developed a lot of tools to do this, but you have to worry about all that. The chemistry, structural morphology, the biomechanics, the degradation profiles, all those things matter if you're going to get optimal tissue outcomes. And I'm sparing you all those details for each tissue type today, so I'm sure you'll be happy with that. But then we take that and we go ahead and cultivate and look at what we get. So just for the white adipose tissue, here's an example of what Roz gets on the right-hand side. And if you know anything about fat, you should be excited to see this, right? Those orange globules are humongous, right? So we've got really good in vitro mature human adipose tissue. That's quite remarkable for those of you who study this in the field. And I won't walk you through all the details, but there are lots of different cell types in here because they all survive from that lipoaspirate. They figure out, they self-select, and then you get a tissue that has what you want with all the different cell types. You can see the scaffold is kind of the purplish blue in the background. I'll skip all that, but the scaffold is there obviously to keep things open and porous so you get good mass transfer and you don't have necrosis in your tissues over time. And we're using these kinds of very functional tissues for all kinds of metabolic studies, all kinds of tissue repair, and regeneration studies and patterning studies, which I won't go through today, but these are all done in collaboration with both Irene, who you've heard about already, as well as Mike Levin in the biology department up on the Medford campus. But what we're trying to do is establish this system as a sort of a physiological reactor that we can perturb in different ways and look at how it responds. And Roz is most interested in things like obesity and diabetes, and with a sustainable 3D adipose tissue culture, you can actually start to make progress in understanding drug effects and how overdosing with glucose and so on has an impact on the structure of the tissue, the size of the globules, and the metabolic profile of what you're seeing. And this is all being fed into models as we make progress to try and understand how to predict the best sort of inputs and outputs for the system. I know somebody asked a question, well, where are immune cells? So as many of you know, whether it's obesity and diabetes, immune cells play a huge role. So we also introduce immune cells into our system as part of the process of understanding how low levels of inflammation affect the readouts that we get. So this is very active, ongoing work in the group. In terms of disease models, really what we're doing, and this is really just started because of the convergence lab that you heard about from Charlotte, is to start to take those 3D tissues, and instead of lipoaspirate, now we use reduction mammoplasty sources, but the same idea, feed the whole material mess into our scaffolding and start to grow tissues, and you can see what we have on the left there. And this work was started both with Victoria's lab as well as a little bit with Charlotte's lab. And we can already show so far with fairly short-term studies, we get fairly stable cell responses, and we start to see metabolic responses, whether we're stimulating with epinephrine or with insulin. So the tissues are responding the way you would expect as a starting point. And then obviously we can now start to introduce cancer cells and so on, and that's the next step in the plans as we move forward. All right, so that was the adipose tissue as a first example of how we go about building 3D tissues. I want to talk secondly about brain tissue because number one, it's probably my most passionate tissue these days. I'm intrigued to always learn more about the brain because we know so little. And so we have a group that's worked on developing a three-dimensional cortical neurological cell model. The current postdocs here, Olga and Yuting, are very active in continuing to improve the model that we already have. And the model was based on something very simple. If you look at the cortical region of the brain at the top, you see a layered structure and you see this really important connectivity among neurons to get proper cell signaling and obviously brain function. And so we recapitulated that design in our tissue model at the bottom. If you just look at the center, we call it affectionately the donut model. And the idea is the gray matter is that sort of porous sponge ring around the outside, and that's where the cortical neurons sit when they're seeded into the brain model. And then the green region in the middle, which is the white matter, if you will, the analogous white matter in our in vitro model, is where those cortical neurons have sent out their axons to connect in the middle and hopefully make a functional tissue model. So that's the system that we initiated a few years ago, and it's continuing to progress very, very well in many directions. And so just a few slides to show you what that looks like. Here's one of those sponges at the top left, and you can get a sense of scale here. This is about 12 by 2 millimeters. The center hole is about 2 millimeters where the axons are going to create the connectivity. You can see what that center gel looks like. Lots and lots of axons here that have been propelled into the center by the neurons growing around the outer sponge ring. And just like before, you have to optimize the mechanics of those sponges, you have to optimize the chemistry, the matrix molecules, all the things I mentioned before. I'm skipping all that. And then in the porous ring on the outside, you see the axons, the neurons sitting there as well along the edges of the sponge layer, waiting to send out their projections into the center gels. And I always like to use this slide because it's instructive of how you have to optimize the matrix you're going to use. If we only use that outer porous sponge for these models, we get really nice cell growth around the rims of the sponge, around the porous domains, but you don't see axons really be projecting into the center regions where there's no solid substrate to hold on to. And if you only use the collagen gel, and this was mentioned a little bit even in the first talk, collagens are wonderful, but they are not very stable materials in vitro culture. They tend to contract over time. And so the cells get in there, they're alive, but they don't really send out projections. So they're sort of in steady state, not doing much. They're not going to give you a functional model. And then if we go to the composite system, which is taking the porous sponge in combination with a gel, now we get the best of all worlds, and we have a functional set of structures we can start to at least pretend look like a brain structure and go on from there. So the designs are really important if you're looking for a physiologically relevant set of outcomes. To the point about time and culture, just to make this point here, we go out about 8 weeks in our initial cultures. Our goal is 6 months. And we can see with the 3D systems, the red, we're able to maintain good viability as well as get good readouts of many markers of neuron activity, whether it's synapse-related, adhesion-related, and so on. So the 3D systems seem to be sustainable for reasonable periods of time. We're busy integrating a blood-brain barrier now with Yuting's work, and that should get us out now to hopefully the 6-month time point, but that's work in progress. So one thing is, do you have the right structure or something like the right structure? Another thing is, can you sustain it? The third is, do you have function? So for a brain, for us, function means some kind of electrophysiological reading, some calcium signaling, some biochemical damage response. And a little bit of that is on this slide. The answer is yes, we see all of that. So for example, if we take the brain tissue right there and we drop a weight on it to simulate brain injury, if you will, we see a change in the electrophysiological recordings with local field potential that we measure in 3D structures. We can actually measure the spiking, and it's different depending on the severity of the damage we apply to the model. So that's encouraging, and then things return to steady state after some period of time. And then also we can monitor the biochemical changes over here, and one of the best indicators or markers is glutamate production. And we see glutamate secretion from the damaged tissue, and again, the level we see depends on the degree of damage and so on, and that's something we continue to look at. So you know, the system seems to be functional, at least at this early stage. One of the things we had to do was make sure we converted our system, which started as rat neurological components, into human, and that we've done more recently. And this is the work of Dana Cairns, who's a postdoc in the lab, and she developed a really beautiful human-induced neural stem cell line that we use in all the models. And you can see the beautiful axon networks that form in the gel center as the cells take shop and do what you want. There's really nice calcium signaling. I'm not going to run the video, but it's there as well. So we have a system now that's relevant to the human condition. That's the point. So where are we going? On the cancer side, these are the systems we're running now. These are the collaborations that we have going, using the tools that I've talked to you about, but now working with other labs to try and bring this to some relevance for cancer treatments. And you know, some of you I'm sure know the severity of some of these cancers. I don't have to tell you that. Glioblastoma on the left and the medulloblastoma. So on the left side, we're starting with cell lines. We're in the 3D system. We have a really nice reporter strain here. So we can see if we compare now our niche system, here we had to switch from just the protein gel porous sponge system to a protein plus hyaluronic acid system to get better readouts for what we think we need to see. And the cells seem to be responding very well, much better in 3D than you see in monolayer culture, as you might expect. And they seem to be doing very well in terms of upregulation in the system. And this is the work we're doing with Columbia here. On the right side with University of Connecticut, we're looking at patient-derived primary cells from resected tumor systems. And here in 3D culture, you can see over a couple of months now, we're getting very good, robust astrocyte response in our systems, indicating that the niche that we've created is supportive of these cells in the model, meaning we start to have a system we can use for screening and so on as we move ahead. And all this work is being done in collaboration with the folks below, but also led by Disha. Disha is a PhD student about to finish in the group this coming year. All right, so that's 2 samples, the adipose tissue and the brain models. The last one I'll just touch on very briefly is our human intestinal model. And this is led by Yin Cheng. Yin is a postdoc in the group. And I don't need to review what the intestine does and is, but it's clearly a critical component in all of our bodies. And so a few years ago, we started a system to generate a 3D human intestine system in the laboratory, taking in mind all the sort of design criteria I've already covered with you. So we wanted something sustainable, relevant, and so on. So we built both a small and a large human intestine, and the design is complex, but I'll just say it looks something like this. So think of the green regions here as a hollow tube, like the intestine, right? But that tube is a spongy structure, and then there's a center hollowed core, so the epithelial cells will line that core, and then there are channels within the sponge itself where we feed in other cell types, immune cells, and then now we wrap on the outside our neurological cells to get innervation into the 3D model. I'll just touch on a few of these aspects today. We also built a model with ridges versus no ridges, so we can simulate both the small versus large intestine, and on we go. All these models were first built with cell lines to make sure we could get things working well, and now they're all converted over to primary cell sources using organoids and so on. Okay, so do we have a system that looks and feels like an intestine? So the first answer is yes. Here is some data to show that. So I'll skip both lines just to say here are sort of tight junction evidence that the epithelial cells on that inner lining of the tube are closely associated, and you get beautiful microvilli forming on those cells as things are cultivated over months. You see digestive enzymes produced as well. The staining in red shows you that. So many of the things you want to see for an intestinal model are viable. The bottom line for an intestine is mucus. If you're not getting lots of mucus produced, you don't have a good model. And so you can see here, we get lots of mucus. Here's a cross-section for the 3D patterned lumen, and you see multi-layered mucus, which really recapitulates what you see in the human condition. And if you care about quantitation, you can see the 3D patterned lumen gives us the most copious amounts of mucus compared to the 2D transwell and the non-patterned lumen. So function in terms of mucus is also very, very prominent here. And then the bottom line for us is if you don't have control of oxygen, you also don't have a relevant intestinal system. And that's what we show you here. We use here reporter bacterial cells that light up depending on the oxygen environment. But the bottom line is we measure this. If we're just using transwells, we get down to, I think it was like 10 to 15% oxygen levels, not down to the level you want with a human intestinal system. If we had the non-patterned system, we can get down to microaerophilic conditions, which is what you want to see. And with the pattern conditions, we get down to all the way down to anaerobic conditions. So we really have a model that has the structure, it has the kind of mucus and other responses, and it has the control of oxygen to allow you to move forward. Ultimately, the best readout is a biological one. So in collaboration with Honorine Ward here at Tufts Med, she's been able to take our tissues now and infect them with one example of an infectious disease. This is Cryptosporidium, which many of you may be familiar with. And it's very gratifying to see that when she infects with the spores, they go through the entire life cycle, and then they can be collected and reinfected into the intestinal model again. So it's a nice example of how these tools can now be used for new discoveries, because now that we have a system like that, we can start to look at interventions. And we're very fortunate we just got a joint Gates Foundation award to start to look at therapeutic treatments to prevent the spores from sitting in the intestine and going through life cycles. All right, so, and then again, we switched over to enteroids now, so everything's done. You get all the cell types in there, so you have immune cells, you have the things you want, and as I mentioned, we've already now been innervating the intestinal model as well, and so we have quite a system that I think is going to be very useful for a lot of studies. So we're using this, and we're just getting started. This is Terence Rowe. He's a PhD student. He's interested in colorectal cancer, and so having a good intestinal model like this, we can start to switch out different cell types in terms of the different kinds of cell sources for these kinds of cancers. And so he's working to collect these cells and build the model, but now infusing the cell types in to look at how the cancers develop. And then long-term, he has some ideas on how he wants to study their responses. So we're excited about this because now that we have the relevant model, we can start to make some progress on the cancer side here. All right, so let me close real quickly just to say this is where I started. I showed you three tissue types that we work on, hopefully get a sense of what we care about in terms of the highlights at the bottom there. And I think this is just the tip of the iceberg. I think the tools are continuing to evolve very quickly, both in our lab and many other labs around the country and the world. So take advantage of them. You know, these are good tools to use for the kinds of questions we're addressing in this symposium. I don't want to forget to I put one slide in to mention Irene's work. So Irene is also in our department on the Medford campus, and she does all the imaging that both Charlotte and I talk about in terms of support for these models. And just to give you one sense here, there's a lot of data here, so don't worry about it, but she can follow for us in a non-invasive way. So no dyes, no perturbation to the system, just non-invasive imaging. She can track the metabolic profiles over time. She can track the structures of the matrices, collagen organization, all the things you care about as you study these models. So the collagens here, here's the scaffold as we start out. This is all with 2-photon emission fluorescence and second harmonic generation methods. But here is the work we published together on the 3D adipose tissue. And you know, you go out to a half a year in continuous cultivation. And if you take nothing else away from that picture, it's how it changes over time. So these are not, you know, one stop, one time, you have a picture of what's happening. The cells go through changes, and you need to be able to capture that, understand where your system is to know what the readouts are and how significant they are. All right, let me close. I want to first acknowledge the NIH because we've been very fortunate. We've had support continuous since 2004 for our P41 center that we run here at Tufts, and our partner organization is Columbia down in New York, Gordana Vunjak-Novakovic. And so the whole center is about what I talked about today, building tools like this and getting those tools out to you guys. So like I said, if there's something we're doing that can be useful, let me know. I talked about 3 of our tissues just in some aspects of what we're doing with them. You can see the list of other tissue types we have actively going in the group. So if there's something there I didn't cover, feel free to talk to me about those as well. I also want to just thank our students. Number one, students and postdocs. I have an amazing group. Faculty collaborators all around the world who I'm very thankful for. We have support from many organizations. I won't go through all that, and obviously from the NIH to do what we do. So I'm very grateful for that as well to keep things moving. And I'll stop there and be happy to answer a few questions. Thank you very much. Great. Yeah, great question. So we haven't looked at that in our model. The focus has been more on mechanical damage so far, and also we're starting some work in collaboration with MGH on, I don't know what I'd call it, but flooding with blood to see how it affects cell functions, particularly the astrocytes. So we haven't looked at seizure-related issues. But certainly if that's something of interest, that could be explored using the model in a way you'd like. But you can condition them, right, with electric shock and so on, I think. I mean, they do that with animals, right? They predispose them. So I suppose you could shock the model and then start to look at response. Yeah. Yeah, great point. We have talked about it in the group. We have done nothing along those lines. This model is very, very new. We just published the first paper last year, and more are coming. So not yet. Happy to chat more and see how to approach those things. Yeah, thank you.
Now, just my ignorance will probably be reflected in this question, but is there any evidence of angiogenesis in any of these models?
So the answer is yes. Again, keep in mind this is all in vitro, so it's a little bit odd, okay? But if you go back to our early work on 3D adipose tissue culture, when we do the seeding like I told you, you can easily find assembled blood vessel-like structures. So the endothelial cells certainly pre-organize over time. I wouldn't argue those are functional blood vessels, but certainly those structures form. And then it's a question, can you hook them up to plumbing so they're relevant? And those are things we do look at and study. So the answer is yes, if you have endos in there. Yep.
You mentioned that one of the overall goals for all these 3D tissues is drug screening, but I'm curious what you think are the major barriers to implementing a drug screen using them. For instance, you have very complex tissue, you might have a lot more issues with kind of the narrow reproducibility you really need for drug screening, tension between kind of miniaturization and still getting the complexity that you need, the length of time to look at chronic effects versus the length of time of, so what are the major factors that are going to help with those tensions?
So it's a great question, Josh. So you hit the nail on the head. To me, the biggest issue is to take the tissue. So I say microfluidics aren't good because they're too simplistic, right, for a longer term screening, but in fact they're good because they're scaled at a level you can hook up and run online. So that's the advantage they have, and that's why it's an easier system to start with. Ours have to be scaled down, but you don't want to scale them down to the point you lose relevance. So it's really, to me, an engineering problem that needs to be solved, which is where can you scale them to still be relevant, and then hook up multiple tissue types to each other, and then to integrate the imaging online, as well as flow through downstream metabolic profiling with mass spec and so on. So there is nothing that can't be done. It's just a matter of the resources to engineer all that into a flow-through online system. And then you can imagine everybody would have their cells harvested, 50 tissue types grown, online screening of every drug, and then you have a database, you're ready to go. So that's the future as I see it. Okay.
Okay, go ahead.
How similar do your tissues remain to the tissues that you're trying to model after several months? So you mentioned that they're very dynamic. Are you able to track those changes so that you know that it's changing in the way that normal human tissue would, or is it diverging?
Interesting question. Do you mean do they age, or do you mean that they look the same as when we started?
Do they continue to look the same as the tissue you're trying to model without continuous alteration?
So I can say for the fat, it looks the same, unless we've overdosed or underdosed by design, so the fat globules shrink or swell. Those are things we can track. But otherwise, I would say those tissues look pretty much the same. It's hard to tell with the others yet, I'd say, too early. Check back in a year and we'll compare notes. How's that? Okay. It's a great question, though.
Well, that's some additional questions, so let's go ahead, Evan.
Cells and tissues live in a pulsatile, stress-stricken environment. You look at that dimension and moving to the right.
Yeah, so again, I had to just cut down today. So for example, the intestine model, we have a whole bioreactor with flow through for the lumen, and it has mechanical flow, so it's like peristalsis. So that's all functioning up in the lab. You can come take a look. So yes, we consider that for every tissue type.
The idea about moving it even further to the right, have you considered about taking hard with your model for the colon because of the destruction? Some of the other stuff actually taking it and translating it back into animals, immunocompromised animals, doing drug screens that way, or alternatively even, I guess just even the drug screens alone would be something that you can do. And also actually the influence of the immune system, because that's going to obviously be hard in the vitro setting. We'll use an immunodeficient animal in that case, it's not a thing. Basically putting them back into the animal so they can actually interact with the environment.
Yeah, it's a really interesting point. When I'll tell one short story. So when we started both the brain and the intestine work, there was never intent to go in vivo with them. It's simply in vitro models. But in fact, we have a collaboration at UPenn on the brain system, not the intestine yet. And it's very instructive from what you just raised, because they wanted to look at repairing damaged brain tissue by implanting good brain tissue. And so we prepare the tissues, we get them down to UPenn, they implant them in the animals, and they all fail. At least the first iteration, and they failed because of immune response, right? And it was very instructive because we had to go back for thinking about transplantation, that's not our goal, but for them, it was their goal. We had to think about how to redesign the donut model to control inflammatory responses at the interface so that you could give the tissue a chance to take. And so we've redesigned for that in mind to see how well it will take in this round of in vivo studies. That's the extent that we've used. There's no reason you couldn't expand in the directions you're thinking about, but I'd rather work with others who want to do that because that's not something we really specialize in.
I think it would work in the cancer setting, certainly, especially if you want to get the animals correctly. Can I ask one more quick follow-up? So with all those silk fibrine scaffolds, are you coating them with anything before you put your cells on them?
Almost always, yes.
Yeah, is it just matrix or?
It depends on the tissue type, right? So in the brain systems, it might be a fibronectin combination with a few other things, but it depends on what you're using, what tissue you're after. Yeah.
One last question.
In your adipose model, have you either intentionally or spontaneously come across malignant transformation?
A great question. Not yet, but we haven't really, I mean, none of the students have mentioned seeing it. It's a great point. It'd be really interesting over time to stimulate and see if you can create this as a spontaneous mutant. I like that idea. Yeah, thank you.
Okay, let's thank David again.